Hello, everyone, and good morning. We thank you all for joining our webinar today. My name is Jenny Ohm, and I'm a Technical Application Specialist here at IVIM Technology. Today, we'll be presenting in a webinar how real-time intravital microscopy is revolutionizing cancer immunotherapy research, enabling researchers to have better insights into cancer metabolism and the efficiency of immunotherapies in animal models. Our presenter, Hayden Kwan, Senior Technical Application Specialist at IVIM, holds a Master's in Life Sciences and has extensive experience in both clinical and preclinical imaging and research. If you have any questions during the webinar, please feel free to ask us through the Q&A box and we'll address them during our Q&A session. Hayden, you may begin whenever you're ready.
Thank you, Jenny. And hello, everyone. Thank you very much for attending today's webinar. I'm Hayden Hyungjin Kwon, Application Specialist from IVIM Technology. So today I will present on the imaging technology with intravital microscope with the title of Transforming Cancer Immunotherapy Research with Real-Time Cellular In Vivo Imaging using Intravital Microscopy. I have prepared this presentation based on the data we obtained from our previous study, followed by the virtual demonstration session. This webinar will proceed as follows. First, I will demonstrate a brief overview of IVIM Technology's intravital microscope. Following that, I'm showcasing in vivo imaging application data performed by IVM, obtained through various cancer metastasis and immunotherapeutic effects in mouse models. And at the end of the webinar, you can see how manipulate the device for imaging the cancer mouse model through a video as a live virtual demonstration. Okay, so a conventional in vivo imaging system, X-ray, PET, or MRI imaging, for example, are started off, but they have limitation because they are designed to get the simple whole body imaging or inferring specific organs operating on a microscopic scale. So, a lack of proper resolution makes it impossible to observe the dynamic cellular activities directly in living animals. To address this limitation, IVIM Technology had been trying to develop the optimal in vivo imaging solution, IVM imaging system, which is capable of visualizing cellular level details. This advancement now allows us to have deep insight into the cellular dynamics, analysis of delivery, and efficacy of the drug in real time. Now, I'd like to introduce our latest optimal in vivo imaging system, IVM CMS3, with dual mode confocal and two-photon laser. This is the most compact intravital microscope in the market with co-optimized integrated software. It has many key features, for example, high usability with easy installation, hands-free maintenance, and all-in-one package. And IVIM is leading the intravital imaging field with all-in-one microscope system with super performance. This single box package enables easy installation, operation, and maintenance. And also it has co-optimized hardware and software, which is integrated ultra-fast polygon scanner, temperature monitoring system, motion compensation, and AI denoising software. So let's take a closer look at these key features. IVM imaging systems are equipped with ultra-fast polygon mirror scanners, speed up to 50 frames per second, leading to capture the dynamics of fast-moving cells like the red blood cells or the green neutrophils flowing inside of the blood vessels in the heart. Especially, this ultra-fast polygon mirror scanner maximizes the motion compensation correction algorithm, which enables high precision of motion artifact by facilitating frame-by-frame compensation. So this is being highly optimized solution for imaging rapidly moving organs, such as the heart and the lung, as it can compensate the X and Y movement artifacts with real-time acquisition of compensated data right away. This video on the left shows the recorded video during in vivo heart imaging, acquired during in vivo heart imaging. So in here, the raw real-time video has high degree of movement, but when we apply the motion compensation, we can directly acquire the high quality and clear images without any shifting artifacts. But without algorithm, only these blurred images in the middle could be obtained due to the artifacts, disturbing the quantitative analysis of the data. So I'd like you to get idea that other conventional in vivo microscope only could acquire these kind of blurred images from the same animal models. In addition to the motion compensation function, to maximize the utility of the video rate imaging capability of IVIM's microscope, we developed an AI software to efficiently denoise real-time videos obtained by our microscope. The outcome has successfully preserved the high temporal resolution of the video while removing the noises. And surely, during denoising procedure, even the small-sized molecules like nanoparticles expressing red signals here are also successfully imaged. So previously, it was very difficult because traditional denoising AI models lose the temporal resolution with denoising. However, we have further developed a more advanced internally trained AI model to speed up the denoising processing more than 50 times. So this work was published in Nature Methods in 2023, and IVIM has an exclusive license for the global market. And lastly, especially with this kind of various apparatus, which we call it imaging windows and chambers, users easily can get highly qualified longitudinal in vivo images from each internal organ. For example, with the dorsal skin fold chamber, we could visualize the cells and evaluate drug activities like anti-cancer drugs after inoculating cancer cells inside of the skin. And toward the end of the webinar, you could watch the surgical procedure for implanting this chamber. And we are also optimizing surgical methods for securing a cranial imaging window to the brain, allowing observation of various dynamics such as brain cells and drug permeability across the blood-brain barrier. So if you're interested in observing other organs not listed here for long-term studies, please feel free to reach out to us for potential collaboration and development for or optimization of this kind of window. Based on the advantages of being able to image almost all organs in a living animal's body at the cellular level, especially thanks to these windows and chambers, IVIM Technology's current biomicroscope is used for imaging various disease models, including cancer models, along with dynamics of immune cells. So as you can see here, especially in the middle, we can visualize circulating tumor cells and T cells that are motile toward cancer cells. In other words, with IVM microscopy, we can not only observe the processes such as proliferation and invasion of the cancer, but also track immune cell responses, also the biodistribution of drugs as well. Okay, so move on to the next stage. So first, I will present the findings of the study where real-time observation of tumor infiltrating lymphocytes, TIL, migrating into tumor tissue enabled us to predict the factors determining lymphocyte motility. To assess the infiltration of T cells into the tumors within the breast cancer animal model, we conducted adaptive cell therapy by injecting fluorescent T cells, representing the red signals here. Subsequently, we tracked and observed the activation of tumor-infiltrating lymphocytes using in vivo microscopy on day 1, 3, 8, and 11. As a result, as shown in the left image, we were able to visualize the activity of tumor-infiltrating lymphocytes for each day. So additionally, through time-lapse imaging on each day, as shown in the right video, we were able to track the infiltration of tumor-infiltrating lymphocytes. Following the previous slide, where we observed differences in activity between the inside and outside of the tumor cells, particularly with significant activity inside the tumor on day 11. So we proceeded to observe a xenograft model created by transplanting human breast cancer cells into mice, which are not of the same species. So in contrast to the allograft model, in the xenograft model, strong activity of infiltration was observed in the third day, followed by a gradual decrease in activity thereafter. Also, as shown, TILs exhibited higher activity in the MCF7 models. It is already known that these two kind of cancer cell lines differ in the expression of estrogen receptor. So for the first time, we could demonstrate that this protein, this receptor, plays a role in regulating the motility of tumor-infiltrating lymphocytes. So therefore, tracking the activity of tumor cells and tumor-infiltrating lymphocytes using our in vivo microscope can be a novel approach to understanding the correct correlation between immune responses and tumors in living animals. Next, I have prepared data showcasing the real-time observation of immune modulatory responses of various drugs targeting specific cancer cells. So these observations were conducted using a range of window and chamber implanted mouse models to study how these drugs regulate the immune cell activity for therapeutic purposes. So here, we inoculated LLC lung carcinoma cells, which express GFP into the dorsal skin fold chamber model. So after a few days, the red blood cells were labeled with the fluorophore and injected into the mouse, as shown in the video with the green color. And this demonstrates that we could observe the growth of the cancer cells and also measure the velocity of blood flow near the cancer tissue. And next step, we also visualize the delivery of pharmaceutical nanoparticles. So we injected a triple negative breast cancer cell line into the dorsal skin fold chamber model, followed by the injection of the red nanoparticles. Subsequently, we conducted imaging at day 2, 6, and 24-hour time point post-injection of the drug. So the nanoparticles labeled in red were observed within the blood vessels surrounding the cancer cells at day 2, at 2 and 6-hour time points. But by the 24-hour time point, we could observe that the nanoparticles are exiting the blood vessels and being delivered around the cancerous tissue. So we could get the insight that this drug had a proper delivery system toward breast cancer here. And in the other study, we visualized the delivery of pharmaceutical bispecific antibody drug. A bispecific antibody drug is an engineered antibody capable of simultaneously binding to two different antigens, often used in cancer treatment to bring immune cells such as T cells directly to tumor cells. So this dual targeting ability enhances immune response precision and also reduces off-target effects, making it a promising approach for more effective and safer cancer therapies. So to explore this, we injected a triple negative breast cancer cell line into the dorsal skin fold chamber model, followed by the injection of bispecific antibody drug. Subsequently, we conducted imaging at 4 and 24-hour time points post-injection of the drug. So this data on the right shows that the drugs labeled in green were observed within the blood vessels at 4-hour time points. But by the 24-hour time points here, we could see that bispecific antibodies actively move toward the breast cancer tissue to be colocalized. So we could get more clear insight here, which says that drug had a proper delivery system toward breast cancer. So when examining the cross sections here, we observed that the drug was significantly more localized within the tumor tissue at the 24-hour mark compared to the 4-hour mark. So this finding suggests that bispecific antibody drug used in this study has a high binding affinity for the target cancer cells. So furthermore, with prolonged imaging, we anticipate being able to ultimately observe the destruction or the decrease of the cancer cells. And next, I present an example using a cranial imaging window implanted mouse model for brain imaging rather than the dorsal skin fold chamber model in this slide. So specifically, we imaged GFP expressing glioma cells both before injection into the brain and on the day 7 post-injection. So the results clearly showed that the cancer cells successfully engrafted and proliferated around the injection site. After confirming tumor growth on day 7, we intravenously administered a red fluorescent nanoparticle drug. So day 1 post-injection, the drug was observed successfully reaching the brain and localizing around and partially within the cancer cells. From day 2, white fluorescent immune cells were seen infiltrating the glioma boundary and penetrating the tumor tissue. So this suggests that the administered nanoparticles enhances immune cell activation and recruitment, thereby modulating the immune response to attack the tumor in the brain. Okay, so finally, let's take a quick look at the result data on the drug which is applying the anti-angiogenic effect. So in the untreated group, the blood vessels were significantly dilated, leading to active cancer cell division here on day 13. In contrast, the drug-treated group showed only slight dilation with relatively less cancer cell growth observed. So as demonstrated previously, the dorsal skin fold chamber and also with the cranial imaging window are suitable for long-term imaging of the same location within an individual, making it a very useful technique for analyzing drug responses within the body after administering various treatments. So using IVIM Technology's in vivo microscope, we continuously optimize methods for imaging a wide range of small animals, including mice, rats, and ferrets, allowing for the unrestricted imaging of multiple organs to see the cancer metabolism or the drug activity. So this not only extends to the visualization of cancer animal models using the dorsal skin fold chamber, but also encompasses various other applications. Okay, let's wrap up the introduction and move on to the next stage, the virtual demonstration session. Today, I prepared the recorded virtual demonstration session to give you the insight with melanoma cancer cell implanted dorsal skin fold chamber model. So I additionally labeled the blood vessels through intravenous injection of anti-CD 31 antibody-based labeling kit. The cancer would express green signal and blood vessel will represent the red signal. Okay, so let me start off with introduction of IVM CMS3. As you can see, the main body features a compact design compared to other devices. Below it, we have the confocal to photon laser controller and main power supply. So in this imaging room, there is a mouse model with a dorsal skin for chamber to be used for imaging. And there is a turret equipped with 5 different optic lenses on the ceiling and also 5 ports to connect the mounting stage, glass holder, and rectal proof to maintain the body temperature stable during imaging with this maintenance system. And using the jog dial, we can move the stage. And I briefly prepared the surgical procedure video for implanting dorsal skin for chamber. So first, I remove all dorsal hair with the depilatory cream, and after wiping it out, just look through the skin to find the beautiful vascular structure on the skin. After that, hold the dorsal skin and place it with placing the 2 frame on each side. And using the 16 gauge needle, pierce the skin through the upper opening and secure it with bolts and nuts. Sometimes it takes some time. So you could hold the skin tight with this bolts and nuts. Okay, so using the small consecutive holes on both wings, stitch the skin to secure it in place. So then pierce the skin through the 2 lower openings and secure the infold chamber with bolts and nuts in the same manner. Now, carefully excise one side of the skin to expose the underlying skin on the opposite side. So keep the area hydrated with saline to prevent drying. And subcutaneously inject the desired number of cancer cells mixed with Matrigel and cover it with glass. With holding the glass with the C-type holder. Okay, next video will show you the in vivo imaging process very soon. Now you are seeing the IVM engine interface featuring a user-friendly interface. So on the main screen, you'll notice the separate displays for the DAPI, GFP, RSP, and the sci-fi channels. And additionally, there are displays for the integrated live video, main display, and window for captured images. To ensure the optimal excitation and detection during in vivo imaging time, I have already adjusted the laser power and the gain value. So by activating the scanner and shutter, we can locate the focal plane until the signals are displayed like this, enabling us to observe the morphology of the tumor cells and blood vessels. Using the jog dial, we can navigate to find the best field of view on the cover glass. So, explained before, in this experiment, the red signals represent blood vessels labeled with anti-CD31 antibody conjugated with the fluorescent dyes from the sci-fi channel. And the green signals representing melanoma cancer cell line. The IVM engine also offers the various imaging combinations to suit different needs. Some of these options include the Z-Stack for capturing images at the different depths, Mosaic for wider area imaging, multi-position for capturing multiple spots, time-lapse for capturing images at specific time intervals, and live video recording. So for this imaging session, we will be using the mosaic imaging mode along with the Z-Stack. So this approach allows us to obtain a comprehensive view of the cancer cells and vasculature across its entire depth in wide field. So to perform imaging over the desired area, we need to specify the imaging spot. So I loaded the Z coordinates to capture images starting from the top to the bottom with the 5 micron interval, and total number of images will be 16. So the imaging depth will be 75 micron. So once the images are captured, we can use IVM Studio, the post-processing software, to stack them to generate a Z-projected image. And here, at the mosaic imaging mode tab, you can set the start and the final coordinates of the region of interest. So in this imaging session, total number of images would be 2 by 2, the 4. So after loading the coordinates is done, you can start the imaging with clicking the capturing key. All the movement of the stages will be automatic. So during the image acquisition process, I will now demonstrate how to post-process the acquired data using the IVM Studio, our software to 2D, 3D, and 4D in vivo data analysis. So please, I hope you to understand that to be respectful of your time, we may not be able to cover every function in this short demo. So if you have any questions, please feel free to ask. So to begin, let's proceed with the post-processing of the acquired data by adding the hive directory folder into the IVM Studio. You can see on the left here. After that, you will have access to all the categorized files. So within the software, you can double click or just click this open key to see the data acquired during the previous imaging session. So you can adjust and improve the quality and visibility of the images, such as modifying the color balance, brightness, and the contrast of each individual channel. And once the necessary adjustment has been made, we can play Z-Stack images with the Z-Stack stamp for better visualization and analysis, and also the scale bar. So this allows you to observe the acquired images in a sequential manner, gaining insights into the cancer's tissue structure across the different focal planes. And also, the Studio software has automatic stitching functions. The data will mosaic information has this activated execute key here. So by clicking this key, all the separate individual images will be stitched together automatically very soon. So without the Z projection function, all the images at the located at the individual focal plane. So we could see the depth information images from the surface to the bottom in wide area region of interest. Additionally, we can utilize the depth-wise acquired data sets for 3D reconstruction. So go to the 3D visualization mode and add the folder again. So double click to open the selected data to build a 3D constructed images. So by making, I'm just hand rotating the images. So by making a few adjustments within the software, we can generate and extract the video clips that provide the horizontally rotating 3D view of the cellular structure and orthogonal view to analyze the interaction between cancer cells and therapeutic molecules if you inject it for visualization. So this visualization method complements the 2D images and offers a comprehensive understanding of the tissue's organization and the architecture. Also, you can extract the recorded videos of the 3D built data. Okay. So I like to share the organized imaging data acquired and post-processed with IVM software. So now you're seeing comparison of two images between control mouse model without cancer cells and the cancer mouse model. So on the left side of the screen, you can see the image of blood vessels taken from the normal mouse model with a 25x lens, providing a high resolution view. On the right, there is a stitched image of vasculature and the cancer cells obtained from the cancer cell inoculated mouse model. And here you could watch the video extracted from this spot surrounded by white dotted sphere to see the zigzag play video from surface to the bottom, and horizontal rotating video to see this imaging spot from different angles. And also you could get 3D built data with only applying the stitching function, as I said before. So without the Z projection function, studio software will stitch all the mosaic images from the individual plane, allowing us to visualize cancer cells and vessels to get a closer look at the localization or interaction between cells or the distribution or efficacy of the drug for your study. Okay, throughout this webinar and the virtual demonstration, we have explored the process of in vivo cancer cell imaging using the IVM chemistry, along with the associated chamber-implanted animal model. So thank you for your attention, and if you have any questions regarding the experimental details or any technical queries, please feel free to ask through the Q&A box.
Thank you, Hayden, for the presentation. We'll now move on to a Q&A session and answer some of your questions. Our first question is, in the xenograft model, there is very low T cells in nude mice. It would not be a preferred model to examine T cell infiltration. Is this correct?
Yes, I know where you're coming from. So to address this limitation, we could use the other normal animal model like B6 wild type or other white wild type mice. So in this case, we have the specific labeling kit to label the T cells located inside of the living body. So in this case, we could label the cells with various colors or the fluorophores excited with the different wavelengths of the laser. So you could get the same result or the wonderful imaging data like I showed you in this webinar.
And the next question is, how do you stain vessels with CD31? Is it a genetic model or just by injection, or is fluorescence tagged to CD31?
For visualizing the vessel, we injected a kit designed using anti-CD 31 antibodies that bind to endothelial cells of blood vessels. So these kits can be customized to match various laser labeling ranges. So in addition to staining blood vessels, uh, uh, we could also provide you the other type of the labeling kits. So if you have any inquiries or if you need any further information, please email us or leave a note during on the survey.
And I think we have time for a couple more questions. So the next question is, how long does the dorsal skin fold chamber last when implanted?
The dorsal skin fold chamber typically remains viable for 8 to 10 weeks. However, this duration can vary depending on the number of cancer cells injected. So injecting a large number of cells, especially those with rapid growth characteristics, may push against the glass, reducing the imaging quality. So if you want to observe over a longer period, it is recommended to inject a smaller number of cancer cells.
And our next question is this technology able to image already established bulky solid tumors?
Uh, with using the dorsal skin fold chamber, we are recommending that uh, imaging or visualizing the tumor or the other cells will be performed within 8 to 10 weeks, like I said before. So if you want to use the bulky solid tumors, we are we are suggesting that inject a small number of the cancer cells and image it within 4 weeks or from 4 weeks to 6 weeks. But in terms of using the for if you are if your study strongly needs to see the bulky like 5 to 10 millimeter in diameter solid tumors, maybe we could use a little bit larger dorsal skin fold chamber for to fit your study.
And our last question is, how long are you able to continuously image in the kidney or lung? How do you do the longitudinal imaging over 11 days?
So in terms of the kidney, we could utilize two surgical methods. So first, you could repeatedly open and close the skin at the every time point for of the kidney imaging. So in this case, you could visualize and get the you can acquire the imaging data during 11 days. But also you could utilize the abdominal imaging window. So you can implant this ring type of window on the skin. So it will last over 8 to 12 weeks. So in this case, you could visualize the kidney for over like 2 months. But in terms of the lung imaging, so we need to open the chest wall with um, with cutting the skin and also the several ribbons. So in this case, maybe the duration of the lung imaging will be 4 hours or the 6 hours. I hope you understand that opening the chest wall is quite harmful to the mouse.
So thank you everyone for your questions. The webinar recording will soon be available on our YouTube channel. And at IVIM Technology, we offer both on-site demos starting from a couple of days to weeks. And we also offer virtual demos through Zoom. We can also test your samples through our CRO research services and provide training for new animal surgery models for long-term or terminal intravital imaging. If you have any more questions or if we couldn't answer your question during the Q&A session, feel free to reach out to us at information@ivimtech.com. We appreciate your interest in IVIM technology and we thank you again for your time. We look forward to seeing you again soon. Thank you.